Okay, is everything okay? Everybody's seeing everything. Everything is visible. Okay, I can then just put like this. Okay, so hello everybody. Welcome back to the Cardioscope Athronet webinar series. This is the last one that is going to happen in the year of 2024. I think this is the 14th or 15th that we are doing at this point. So hopefully we did a good job in the past year bringing you this series. I think that the number of topics that we addressed are pretty versatile, and we will continue with that approach in the next 2 years. So to go back to the main topic of today's webinar, it's basically focused on RNA sequencing, on basics of RNA sequencing. I will try to cover mostly basics, as the title says. In some points, I will address certain specific issues that might raise during this process, and also I will try to give you our experience from the things that we have done in previous years, so you can also build maybe your own protocols and approaches based on that. I will just remind you that this is mostly basic stuff that more advanced lectures have been previously addressed. We have a webinar that was led by Matthias from Tamirna. He addressed mostly sequencing that is focused on RNA sequencing that is focused on small RNA, microRNAs to be exact, and other one is about circRNAs that was given by Fabio. So at some point during the lecture, I will refer you to those two webinars because they are mostly focused on super specific stuff. And here we are just discussing in general the principles of RNA sequencing. So let's begin. Let's see what actually is the transcriptomics and what RNA sequencing is. So basically, so far, there are many different types of RNA sequencing. Of course, with the advancement of next generation sequencing, first being established on DNA and then later on transferred to RNA, the technology evolved pretty quickly and find applications in many different things. What today we will discuss is mostly focused on bulk transcriptomics or bulk RNA sequencing, when basically you have the entire tissue, entire sample with different types of cells. You destroy it, you extract RNAs, and then perform sequencing. And basically, you can this type of analysis can give you information about differential expression genes in this tissue, but still there are some specific information missing. For example, in which particular type of cells these transcripts are enriched or differentially expressed. And this is why in the last 5 years, I think 5, maybe 7 years, there is more and more papers going into single cell transcriptomics, where basically you combine single cell technology where you divide cells and then perform transcriptomic analysis, and then you get more insights into the distribution of particular transcripts or genes within certain cell types or cell subtypes. And then this led to another application, which is currently, I would say, maybe most expensive and also the latest evolutionary advancements of the technology. It's spatial transcriptomics, where basically you combine spatial approach where you have with transcriptomics, and you get the information of particular genes and their enrichment in specific parts of the tissue. And this can really be of great importance in atherosclerosis, when you are looking at atherosclerotic plaques and you want to see particular genes being super expressed, I don't know, in the part that is maybe vulnerable, or on the shoulders of the plaque where where there is not so much inflammation going, and things like that. And as I said, the focus of today's lecture is going to be bulk transcriptomics. Maybe I will try, because it is also in my interest to hear lectures about single cell transcriptomics and spatial transcriptomics. We will try to find people who are really experienced with this, and maybe in the next year or two, uh, um, bring them to the webinar series to give us more insights on the possibilities on these technologies. So, of course, everything starts with the proper pipeline and proper workflow and the setup design. So this is just an overview of all the things that are involved from the beginning to the end when we discuss RNA sequencing. So in the beginning, we have experiment or study, which of course designates which type of cells on which type of materials we have available. Then we go to the preparation. Several steps are needed to be taken before actual sequencing. We will discuss this. Then you go into actual sequencing. Then you get a lot of data. Then your actual headache starts at this point, because you need to know what to do with this data and how to comprehend the data. Today I will not be going into bioinformatics, because I'm not very experienced at this in the bioinformatical analysis of the data, and hopefully at some point we will have also opportunity to reprise our lecture that we had in the training school that we organize in Crete. I also asked Matthew to join us today, but unfortunately, due to his overwhelming obligations in the final PhD years, he was not able to join us. But probably at some point next year, he will be able to give a lecture about bioinformatics and how to perform extensive analysis of the data after RNA sequencing. I would start with basic terminology behind all this. This does not really apply only on RNA sequencing. It's also something that the jargon is used in DNA sequencing as well. But I will just quickly try to summarize all these terms for you, because I think it is important. For me, it was quite a conundrum when I was start reading about RNA sequencing and getting deeper into the logic behind the technology, to really try to understand the difference between many of the terms that are frequently used and sometimes interchangeably used also, although they are not really synonyms. So first, let's start with a term that is called read. Read is basically sequence of nucleotides that is generated during the sequencing, and we will many times mention this term read, number of reads, which basically means you will see when we discuss how the protocols are done. You basically destroy your RNA, at least in Illumina sequencing, and then you get many of the fragments of your RNA that is going to be in Illumina sequencing converted to cDNA, and then you sequence these fragments, and every signal that you get, every fraction of the signal is basically read. Next, I want to give you a little bit of information about single-end sequencing and paired-end sequencing, although the paired-end sequencing nowadays is becoming more and more popular. It is a little bit more expensive, I would say, but it gives you much more information and much more certainty to your data and to your process. So basically, as the names are suggesting, single-end sequencing, it basically involves sequencing just from one side, one end, and paired-end sequencing is sequence your particular sequence of interest from both sides. So you get two information per one sequence, which of course increases the precision and accuracy of the sequencing. Read length is basically the number of nucleotides in each read generated by sequencing process. And currently, with the Illumina sequencing, most of the time you get transcripts that are between 100 and 300 nucleotides long. Alignment is another term that is frequently used in this terminology. And alignment is basically a process where you align what you get from your sequencing data to the reference genome or transcriptome. And as you can see here on this figure, I don't know if I can just, yeah, this is the pointer. You can see that basically during the sequencing you get a lot of reads, and these reads are basically parts or specific parts of specific genes, and then you need to align these sequences to your genome, so you know that these reads are coming from this transcript or that transcript. And reference genome is a representative example of a species complete set of DNA, which basically means it's some kind of standard DNA sequence that you can align your genome to or map your genome to. Now, two terms that are also very frequently used, especially if you plan some of your RNA sequencing experiments in the future. Somebody might ask you, so what type of coverage you need, or what type of what depth of sequencing you aim to have. And this is why I think it's very important to understand these type of things. They are interconnected, but they are not synonyms. So in this way, I would like to explain to you, at least from my perspective, what is the difference between coverage and depth. So basically, coverage is give you the answer to the question, did you sequence certain particular region of the gene? And depth gives you the information, how many times did you sequence that particular region? So of course, the greater the depth, the greater the coverage, but it is not, it shouldn't be used as synonyms. And I think you can see on the next slide, on the next figure, I think it will be much more clear. So imagine this is the gene of your interest that you want to sequence. And then in order to sequence the entire gene, if you sequence the entire gene, then you will have total coverage of sequencing of that gene. However, in this case, you see that certain reads coming from this gene are covering with more depth certain part of the gene. For example, if you look here, you will see that 1, 2, 3, 4, 5, 6, 7, 8 reads are covering this part of the gene. So this part of the gene, the depth of this sequencing is 7 times, okay? And here, if you go to this part of the gene, you will see that only one read is covering this part. So this part is covered with depth of 1 time. If you concentrate your attention to this part of the gene, you will see that it's not covered at all. So coverage always is a term that is a little bit gray zone and a little bit elusive, because, for example, this gene, particular gene, if you look, if this would be real sequencing data, you would say you have super high coverage, because almost the entire gene length is covered with at least one of the reads. But if, for example, you are super focused on this region of the gene and you want to say, I want to study this particular part of the gene, then you would say that the coverage is bad because none of the reads are covering that area. On the other hand, you can see clearly here that depth is very important, because if you have more depth, then you are more certain that this sequence is present in your sample. And now I want to show you the example why it is important to understand the depth of sequencing and how depth of sequencing correlates pragmatically, practically, with your setup when you're with your experimental setup. So why depth is important. For me, the depth of sequencing is basically your ability to find something in your sequencing data. And this is maybe very plastic example, very artificial example, but I think it will give us enough understanding how it works. Imagine that your sample has only 4 transcripts of interest, okay? And then you ask yourself, how many times I need to sequence this RNA that I extracted in order to make sure that at least I sequence every of these transcripts once. And I always like to use kind of a real life example. So for me, it's like having a bag and trying to reach for the certain objects in the bag. So imagine that you have 4 objects in the bag. Of course, I put this on the slide, woman's bag, and women try to reach. For me, women bags are always quite a mystery and conundrum. I never tend to find anything in them, and then my wife comes and finds everything in them. But just for the purpose of this example, let's imagine that I can actually find something in a woman's bag. So imagine I have a bag and I have 4 different things inside. So the question is, if I want to pick up all the 4 things, how many times I need to reach in the bag? And the question is, the answer to that question is 4. So I need to reach 4 times in the bag in order to pick up 4 different things, and that's supposed to be easy. However, imagine this is kind of more realistic scenario with women's bags. They tend to have many more things, not just 4. And imagine that you now need to, so for example, there are, I don't know, 4 different types of candies in the bag. Okay, there are blue, green, orange, and yellow, and the bag is full of these type of candies. And the question is, how many times you need to reach in the bag to make sure you pulled out every type of the candy. So the blue one, the green one, the yellow one, and the orange one. And if you say 4, then you will be completely wrong, because just based on the statistics, you can do this experiment at home. It's completely safe. And you can actually try to do this. You just use, I don't know, if you have Legos or something, and you try to pick up with four attempts to pick something. You will see that you will not, I mean, there is a very low probability that out of 4 picks, you will pick 4 different things in a row. There is much greater probability that you will pick more, I don't know, 2 greens, 2 blues, and 2 blue and 1 orange, and then you will miss the yellow one. So this is completely correlates with the depth of sequencing, because this is a scenario where you have many transcripts in your cell and you are asking yourself, so how deep I need to go in order to really find these targets and find them in my data, because you need to make sure that your transcriptome is equally represented in your data, because you don't want to miss any genes. And in this scenario, 4 times, as I mentioned, is not the greatest. It's not the correct answer. So you can ask me how many times you need to go. I'm not a mathematician. I always try to run away from mathematics, so I cannot tell you the correct answer, but I know that it's certainly not 4. Anyway, imagine now that this is your cell. These are the transcripts, but you are really interested in this purple transcript or pink transcript that is relatively low expressed. So how many times you need to reach in the bag to pick up the purple or the pink candy? For sure, a lot more than to pick any of these, right? Because it is really, statistically, it is because this is, you are not targeting anything. You are just picking things from the bag. So it takes you quite a lot of attempts to pick up the pink one. And this is actually the same with RNA sequencing. So the lesson that you need to learn and take home message from this is, if you have less abundant targets, then you need to sequence deep, or you need to increase the depth of sequencing. And this is what you will see in real life. For example, if you want to do basic gene expressions in tissues, just to see differential expression, you can go with a couple of millions of reads. I don't know, 5, 10, 15 millions of reads. If you want to go, for example, and analyze long non-coding RNAs that are relatively low abundant, then you need to increase your depth of sequencing to sometimes even hundreds of millions, but let's say 50, 80, 100 million, and so on. So if you are interested in super low express targets, then the sequencing depth is going to be increased. Of course, with the increasing the depth of sequencing, the price of the sequencing per sample increases substantially. That's something to be kept in mind. So before we start with the sequencing, we need to really make sure that we understand what we want to do and what are the prerequisites or predeterminants of our samples. So we know that when we do sequencing, when we perform sequencing, that the data that we have are reliable data. So the first step, of course, is going to be sample collection, and type of sample will really can influence the approach to sequencing that are you going to use. For example, if you have cell lines, because most of the time they are super activated, their transcriptome is pretty rich, you will have relatively good expression, and the number of genes that you can detect is much higher. So maybe you will not need to go super deep with that type of sequencing. If you have blood, with full blood, again, because you have many cells that are going to be destroyed in order to extract RNA, again, super deep sequencing is not needed. If you have plasma, then you will need to go very, very deep, or you need to enrich your sample very much, because, of course, in plasma, there is not too much RNA. Also, if you have tissues, depending on the tissue, you will get the different challenges. For example, if some of you had luck to work with atherosclerotic plaques, it is very hard to extract RNA for atherosclerotic plaques. So I think that also contributes to the challenges when you want to do RNA sequencing. From one side, plaque is very, very rare sample, and it's very precious. And then from this type of sample, you get very low amounts of RNA. And then again, you need to think about sequencing that you are going to do. So, for example, if you have some plaques and you want to do some sequencing, I recommend to go a bit with a bit greater depth of sequencing, because with low depth, I don't think that you can get too much. Then there are RNA extraction methods that will also influence the final output. So because sequencing is so specific and so I would say under the influence of many factors, you want to have samples that are very, very good quality. So most of the time, what is suggested is to use RNA extraction methods with columns, and also to use DNA step to clean the DNA so you don't have the contamination. And then after you extract your RNA, you need to check for the quality, first the quantity, of course, and for the contamination, classical UV analysis, but also what needs to be done, it's to check the integrity of RNA. Now, I don't know if you have any information how to check integrity. Previously, many years ago, we used gel electrophoresis, but now we use basically capillary electrophoresis. Agilent has instruments for those. And then you can basically run your RNA samples, and based on the integrity of samples of your RNAs, you will get a chromatogram that looks like this. Here you can see the perfect chromatogram with RIN, or RIN number around 10. RIN stands for RNA integrity number, which basically compares the areas under the curve of 20S, 28S, and 18S ribosomal RNAs. And calculate this number. If the sample is being destroyed from some reason, for example, the clinical samples have been left on the bench and you didn't have time to process it in a optimal timing, then it will cause the destruction of RNA, and then you will see that the peaks of 20AS and 18S are a little bit less sharp, and then you get increase in these smaller RNAs here, which basically suggests that you have destruction. When you ask people, so what type of free number is required for the sequencing, they will always tell you above 9, but in reality, above 8 is good enough for sequencing. Below 6, I wouldn't go and spend your money on those type of samples when it comes to sequencing. Next question that you also need to ask yourself before actually going to sequencing is, what is your target of interest? Because when you extract RNA, if you don't do certain enrichments during the extraction, you have total RNA. Basically, this is a mishmash of different types of RNA. Unfortunately, these different types of RNA are not present at all at equal amounts. Ribosomal RNAs are basically 80% of total RNA, and left around 15% is tRNAs, and then all the other RNAs, all the mRNAs, microRNAs, circRNAs, lincRNAs, everything goes in this 5%. So just to put that in perspective, if you have 1 microgram of RNA to start, 80% is going to be, so 800 nanograms is ribosomal RNA, 150 is tRNA, and basically 50 nanograms is what you have left for the analysis. Of course, ribosomal RNAs most of the time are not of interest to researchers, so they need to be removed. Now, the question is why we need to remove these ones. Well, just to go to remind you a little bit to the previous metaphor I used, if you have too many transcripts of one type, you need to go very deep in order to catch those that are very low abundant. So in this graphical representation, these blue lines represent ribosomal RNAs, and then these here, other colors are different types of RNAs. So what you can do, you can use total RNA, of course, for sequencing and go very deep, but then maybe it is better to somehow remove ribosomal RNA or enrich specific type of RNA in order to not being forced to go super deep with sequencing, but again, gaining good results. So if you are in RNA business, the chances are that you are not super interested in ribosomal RNA. So what are the two main approaches that we use to select those RNAs that are of our interest? If you are mostly interested in mRNA, then there is one structural feature that you can benefit from, and that's poly A tail. We know that in eukaryotes, all mRNAs have poly A tails. So you can select those RNAs that have poly A tails. This will provide you transcriptomic data set that basically preserves all mRNAs, and also some lincRNAs, because some of them have poly A tails, but you will lose at least 50% of lincRNAs. You will lose microRNAs, circRNAs, and other very interesting type of RNAs. And this process is called poly A enrichment or poly A selection. And it's based on the use of specific oligonucleotides, which are poly T nucleotides, which are bind to some type of either a magnetic bead or streptavidin or whatever. And then you can basically make these duplexes, and then you can select and pull out of the solution those that are of your interest. Other approach is basically to destroy ribosomal RNAs with process that is called ribodepletion, where you basically select only ribosomal RNAs. I will show you in a second how it's done, and then destroy them. And what is left within your sample are all the other types of RNA. So with poly-A tailing, with the poly-A selection, there are several approaches, but they're all based on the similar principle. You have beads, either magnetic beads or streptavidin beads, that have poly-T oligonucleotides. And then when you mix it with your total RNA, there is going to be interaction. Then you use magnetic force to pull out of the solution poly-A RNAs, which are mostly messenger RNAs. With ribosomal depletion, there are two different approaches. We tried both in our lab here in Luxembourg, in Ivan's lab. One is basically based on the use of oligonucleotides, DNA oligonucleotides that are specifically recognizing ribosomal RNA sequences. So you mix everything together. There are duplexes between rRNAs and these oligonucleotides. Then you expose these complexes to RNase H. RNase H is an enzyme that is specifically destroying RNA within RNA DNA duplexes. And this is what is represented here. And then what you are left with is your RNA of interest and a lot of these oligodNA sequences or fragments that you need to destroy again. And then you need to use DNase 1 treatment to destroy DNA. And then what you're left with is basically a ribo minus RNA. So everything will be except ribosomal RNA within your sample. Of course, many cleaning steps are needed between these, which really compromise the quantity in the end that you can get. With other approach seems a little bit more straightforward. It uses basically magnetic beads that recognize magnetic beads or streptavidin biotin system, whatever, that binds to specific probes that recognize riboRNA, and then you just pull the RNA, ribosomal RNA out of your sample, and what you are left with is basically everything else. We tried both approaches. For me, this first one that I described works a little bit better. It's a little bit easier to work the product. I mean, the chemistry itself, when you handle in the lab, it's a little bit easy to do. It's not so complicated. And with this one, to me, it was quite a messy thing to do. The only problem is that on the market, with this one, you have only regions that can do the RNA depletion for the quantity of RNA up to 1 or 2 micrograms. And for this one, you can go up to 10 micrograms. But it is a little bit messy, at least for me. So we can now dedicate ourselves a little time to actually how sequencing is done. So these are all just kind of introductory parts that I think are important to have in mind when you are thinking about doing RNA sequencing. Now, currently on the market, there are 3 technologies for sequencing, and we covered this also in the lecture last January that Rizien from University of Malta presented us for DNA sequencing. So we have Illumina sequencing and PacBio that are based on the principle that is called sequencing by synthesis. And we have completely different technology that is called direct RNA sequencing that is developed by Oxford Nanopore Technologies. So here on this graph, you can clearly see difference in steps with basically direct RNA sequencing. You have very simple protocol. You have only adaptor ligation, and then you do the sequencing. With 2 other approaches, when you have sequencing by synthesis, you have fragmentation, you have cDNA synthesis, you have adaptor ligation, PCR amplification, and if needed, size selection. And then this black line here is basically representing Illumina, and this green line here is presenting PacBio. system. But basically the system, how it works, the sequencing itself, it's done on the same way, in the same way. It's just that the setup is a little bit different. So here you can have more longer reads, and here you have shorter reads. But both principles are sequencing by synthesis, and both require transformation of RNA to cDNA. Only currently on the market, the only approach that you can use to directly sequence RNA is Oxford Nanopore technologies. And I will show you, because I'm not really familiar with PacBio technology, I will explain you Illumina type of sequencing and Oxford Nanopore sequencing. So in Illumina-based sequencing, you have several steps. First, of course, you have the extraction of RNA, then you have the fragmentation. And this is really important because in Illumina sequencing, you don't have long reads. This is very related to the capacity of DNA polymerase to work efficiently on shorter fragments. With longer fragments, it tends to lose the accuracy. And then you convert these short fragments to cDNA, and then basically you do the DNA or cDNA sequencing. One additional step is needed. It's basically ligation of specific adapters, and I will show you why this is important to have specific adapters. In this step, you can also do multiplexing. Multiplexing means that you basically can combine several different samples in one library, and then later bioinformatically you can make the differentiation between every of these samples. So if you have 5 patients, you can pull them together, but before pulling, you add specific adapters that to the sequences that will in bioinformatic protocol tell bioinformatician, okay, these reads are coming from sample A, these are coming from sample B, and so on. And then you perform NGS, and in the end, of course, you do extensive bioinformatics. So let's try to understand how Illumina RNA sequencing is done, or basically cDNA sequencing. So as I mentioned earlier, first steps are converting RNA to cDNA and ligating certain adapters. Now, why these adapters are important? They are important because of the next step. And in the next step, you have what we called bridge amplification. So basically, flow cell is the part of the instrument where the sequencing is happening. It's a basically glass surface, and on this glass surface, you have attached many of these short adapters. And these adapters are going to be recognized by the adapters that you put on your sequence during the library prep. So in order for your cDNA to stick to the flow cell, you need to have perfect match between these adapters. So why we need the bridge amplification? Why we just don't end process here and start sequencing? Well, the point is, if you start sequencing at this point, you will get very slow, very low signal that is not going to be efficiently read by the instrument. So what you need to do, you basically need to amplify artificially every of these cDNA sequences that are coming from your sample. And here you can see how it's done. Basically, your sequencing is stuck to the flow cell through the interaction of adapters. Then instrument promotes this looping or folding of DNA. Then there is a primer binder. Then you have reverse synthesis of the reverse complementary strand. And then basically what you get at the end of this process is 2 copies of the same fragment. And this is extensively repeated numerous times in order to get these clonal copies or clusters. So what you basically have here, it's sequences that are coming from only one type of sequence from your sample. And now when you start sequencing, you will get the same signal on every position for every of those in theory, of course, for every of those molecules that are bind to the surface, and then you're basically just amplifying the signal itself. And this is what is happening in the next step. After you have the bridge amplification and you create clusters of your specific cDNA sequences, then you go to sequencing. And sequencing basically is reading the sequence during the process of synthesis. How it's done? It is basically classical DNA polymerase based activity. So you have the primer in your mixture. You have nucleotides, and every nucleotide is have specific fluorescent dye attached to it. So what it happens when DNA polymerase start to attach nucleotides to the certain sequence due to the complementarity rule, then you will get the fluorescent dye will going to be released, and then specific frequency of light you will be able to record by the instrument, and then you get this signal telling you that on this particular position you have T or G or C or A. Because they are all of different colors, you will get different signals. Of course, because this is based on the DNA polymerase activity, this is why we need to transform the RNA to cDNA, and also because DNA polymerase is not perfect, there are certain levels of mistakes that are happening at this point, because sometimes DNA polymerase, for no particular reason, just based on the statistics and its capability to do its function, can make mismatches, and then you will get certain mistakes in your sequencing data. With Oxford nanopore sequencing, there is completely different story. There is a pore, which is protein pore, embedded in the membrane, and there are a lot of ions and strong ionic current within going through the membrane. And another important player in this story is the motor protein, which is going to be attached to your RNA. And then basically this motor protein is helicase. We know what helicase does. It basically during the replication, it tries to unwrap the DNA. And here you can see that this helicase will specifically bind to this protein and navigate the RNA through the pore. Now, what is happening here? Because there is an ionic current while the RNA molecule is traveling through the pore, you will have changes in the ionic current, and this is going to be recorded as signal. And the good thing is that every single molecule, every single nucleotide has different imprint within this signal, which means that if you have A, it's going to give you different disruption of current. G is going to give you different disruption of current. And also, if you have modified bases, so non-standard bases like M6A or N6-methyladenosine or inosine or anything that is unordinary, again, you will see different in the signal. And from this, you will be able to basically understand what is the sequence when you decode the signal, which is again done bioinformatically. Just to be clear, ONT developed this approach for DNA sequencing. And the original intent of this technology was to enable long DNA reads, okay? But it's also at some point adopted to RNA sequencing. Now, RNA sequencing, we call it direct RNA sequencing, right? But you will see that in this protocol, we also use reverse transcription. It is not necessary, but it is highly recommended to you do reverse transcription, and I will explain how this helps and why it doesn't affect direct RNA sequencing. So basically what you do in the beginning with your total RNA, you basically bind specific type of nucleotides that are recognizing poly-A tails. So if you just start with total RNA with ONT classical approach, you will basically do poly-A selection within the library preparation. Why? Because only poly-A mRNAs are, or not mRNAs, but poly-A RNAs are going to be recognized by these oligonucleotides, and then everything else will basically not survive to the sequencing point. When you do the cleanup, because after this step you need to do the cleanup, everything else will going to be destroyed. You will only have poly-A nucleotide, poly-A transcripts in your library. We wanted at some point to optimize this method to go with ribosomal depletion and then to do poly-A tailing by ourselves. So everything that doesn't have poly-A tail gets the poly-A tail, and then you have much more rich transcriptome to analyze. Unfortunately, because we work with clinical samples and we have very limited amount of RNA to start with, we couldn't do it because for this to work, we need around, I think, 7-8 micrograms of RNA. And for most samples we have is around 1 to 2 micrograms. So not to digress too much, I want just for you to realize that this type of sequencing in its original form, without any modification, is poly-A dependent and focuses only on poly-A transcripts. After you add this, you do reverse transcription, and then you get RNA cDNA hybrid. Now, you can clearly ask me, okay, Miron, why you do this? Why we need this? Because if it's direct RNA sequencing, why we convert it to cDNA? In this particular methodology, it is not the point that we want to convert RNA to cDNA. Why? Wow, but why we do reverse transcription? Because we know that RNA are single-strand molecules that tend to have many secondary structures, and if you have secondary structures, they will be much more reluctant going efficiently into the pore, and then the efficiency of your sequencing is going to be decreased. So when you basically do a reverse transcription, you create this transcript that is combined, this duplex between RNA and cDNA that is linear, and then in the next step you adapt, you basically ligate adaptor protein, and then you perform sequencing. So this adapter has motor protein. It is attached to your hybrid, and then it navigates the double-strand hybrid into the pore. But again, keep in mind that the helicase is designed in a specific way, so only RNA molecule in this duplex is going to pass through the pores. cDNA part is going to be released in the solution, and it's not going to be sequenced, so not in any way it interferes with sequencing. How it's done? Because for DNA sequencing is the entire concept behind DNA sequencing is to start from 5' end to 3' end, and in RNA sequencing they start from 3' end and then with 5' end. So with this, there is no way when you are doing RNA sequencing to actually sequence this double-strand DNA, and it will in no way interfere with your data. After that, you basically get a lot of signals, a huge amount of data. I think I mentioned this several times during the training school. For example, we have the database for 200 samples that were done with Illumina sequencing for microRNAs, and average depth was around 30 to 50 million reads, and we have 200 gigabytes of data for that data set. And here, for my postdoc, I did around 46 samples. Average depth was around 2 million reads, and I have around 4 or 5 terabytes of data coming from this. So the amount of data that you create with Nanopore is much, much more extensive in terms of everything you can imagine. But one of the perks of RNA sequencing, of direct RNA sequencing, is ability not only to analyze canonical databases, canonical nucleotides, but also to be able to identify modified bases. Currently, nanopore sequencing enables you to efficiently and accurately determine the positions of M6A and M5C, and they are currently, I think, developing pipeline to be able to recognize inosine. Currently, it's still not official, but I think next year is going to be publicly available. Now, we know that RNA modifications are implicated in numerous functions, not only on RNAs for a long time, but also for mRNAs and non-coding RNAs and microRNAs. We have wonderful webinars that Yale gave us last year. It was the beginning of this year, actually, about how these modifications are very relevant in microRNA biology and their functions. So this is why Nanopore, it's really, really nice because it allows you to basically collect in one run all these different layers of data, and then, of course, analyze it. And then I will just briefly go into the concept of normalization, although it is not per se wet lab part, but just to give you kind of brief information why it is important for you to try to understand the concept of normalization. Normalization is this, it's similar. If you did qPCR, you know that basically most of the time normalize gene expression of gene of interest to housekeeping genes, or to be precise, endogenous controls or reference genes. And this is a similar approach needs to be taken here. So why we do the normalization? Imagine the scenario where you have three different samples. You extract exactly the same amount of RNA, and then you perform library preparation and you do sequencing, but you end up with different number of reads in all these samples. For example, in sample A, you get 10 million, in sample B, 20 million, and in sample C, 30 million. And this is reality. No matter how good your protocols are, you will never be able to say that out of 100 samples that you sequence, you will have the repeatability of depth in all these to the exactly same number. They will always, when you do this, when you ask company, I want to do this, they will always say, yeah, the sequencing depth is going to be between 50 and 80 million or 80 and 100 million, something like that. They will never be able to guarantee that every sample will have the same depth or the same depth. So in that sense, it is very, if you, for example, do not normalize to number of reads, then what you will conclude is that gene X in sample A is less expressed in gene B and less expressed in gene C, just because you have less signal coming from these samples respectively. However, this is not due to the fact that genes are differentially expressed, but to the fact that you did sequence sample A with lower depth in comparison to sample B and C. The other important thing to take into account is the fact that some genes are quite long, some are quite short, and this will also influence the results that you can get. For example, if you have short gene, then you will have shorter amount of reads that are covering this gene, and if you have longer gene, you will have more reads that are covering different part of the gene. So in this example, you have gene A when there is 1 kilobase, and GB that is 2 kilobases long, and then this one is covered with the depth of 5 times, and this by the depth of 10 times. And then if you just compare the signals, you count number of reads, you will say, gene A is less expressed than gene B, but this is not the case. It is just that we have much more reads coming from the gene B because it is longer. So to cut long story short, most of the times when we do the normalization, what we do is basically express our results as fragments per kilobase of transcript per million of med reads. In this way, we normalize to the length of gene and also to the number of reads in every sample, and then we basically get much more standardized number that can be compared in between samples, and then you will actually get a much more relevant conclusion. And for the end, I just want to put out, this is a little bit hard to digest. I just put this as an information on when you compare these two technologies, what would be pros and cons for one or the other. For Illumina, I think high accuracy is still something that is pro. The pipelines are much more developed. There is higher throughput. Per sample you will get, or per read you will get less prices. So these would be the pros for Illumina sequencing. But on the other hand, you cannot do direct RNA sequencing. So if you are interested in epitranscriptomics or long reads, Illumina sequencing is not the way to go. For Oxford Nanopore, major minuses are low accuracy. It's still not good as Illumina, although it is improving dramatically from one year to another. Also, it is easier to start with this one because the instruments are not so super expensive, but then the chemistry is, so when you calculate the per sample or per read, it's going to be a little bit more expensive. But on the other hand, it really gives you a lot more information than Illumina sequencing. With Illumina sequencing, you cannot really have information about isoforms, not without a super developed specific pipelines. And here with Oxford Nanopore, because they are longer reads, you get really nice information about the isoforms. You can also get the information about different modifications. Currently, as I underlined, it's just M6A and M5 cytosine, but in the future, it's going to be also, I think, inosine is going to be one that they are currently developing. They are working on. Of course, there are some pipelines that are not officially from the company to also try to find inosine in your data, but they are still not officially developed. It's from the side of the company. Also, another thing that is also good for Oxford Nanopore is basically you do your sequencing, you finish your analysis, and then 2 years later they develop the pipeline for some new modifications. You can go back within your data and find those modifications within your data set through new pipelines, because data is already there. These signals that are different between different modifications and different nucleotides are all already in your original raw data. But you just need new pipelines, bioinformatic pipelines, to understand how these signals differ between each other, and to be able with great accuracy to say, okay, in this position, this is this modification and this is that modification. And of course, keep in mind that this technology is still in development. It's still in this early stage, at least with the RNA. It's not so, it's much more developed for DNA. For RNA, it's a little bit less developed, but I really hope that they will invest a lot more efforts developing new RNA technologies and approaches for RNA analysis. And with that, I'm finishing this one. This is a nice picture from the summer school, for the training school that we had in Crete, just to warm us during these cold December days. And also additional point for all you younger guys out there, we are currently working on the book that is basically that will focus on transcriptomics in atherosclerosis, and all these information that we are providing you through webinars are going to be basically compiled in this book, and it should be available sometime next year. Of course, you will get more information about it. I just wanted to give you a heads up what we are currently working on. So that's from me for now. I hope I didn't confuse you. I hope I gave you a lot of a good explanation, so you understand the basics behind this approach, so you can at least understand the basic concepts and then build your knowledge after, and really understand how you can apply specific protocols for your purposes. And I'm open to any questions, of course.
Thank you very much, Miron. That was an outstanding webinar regarding the basics of RNA sequencing. It was great. It was really great. Perhaps we can ask the audience for questions. Just one question from me, very simple question. Now, when we're speaking about depth, I understood that in Illumina technology, we need a lot of depth because we need to sequence many samples because they are broken into many pieces. But in Oxford, does this apply also, the depth? Because here we do direct RNA sequencing, and we don't have, disruption of the RNAs. The depth should be different, right?
It is not comparable. You cannot say, for example, 5 million of reads in nanopore is comparable to 5 million reads in Oxford in Illumina sequencing. That's for sure. Maybe there is somewhere scale how you can translate one to the other, but for sure with direct RNA sequencing you always get less depth than with Illumina.
But you need also
from the same amount of starting RNA, because at some point in Illumina you have the cDNA, you have the amplification, you have the PCR reaction. So you need less amount of starting RNA, and then you expand this through the amplification, and then you get much more depth. For example, I didn't go into these details. I didn't want to confuse the audience during the lecture, but you can also do this with Oxford Nanopore. You can do transformation of RNA to cDNA, then you do PCR, and then you do classical cDNA sequencing. In that sense, you will lose everything that is coming from the modifications side, but again, you will get longer reads and you will be much more accurate with the isoform analysis. And then, for example, in that case, I saw some papers where they did this and they analyzed the RNA from plasma not directly, but through amplification, and then perform the cDNA, the DNA sequencing and they go with 80 million reads and things like that. For example, currently what we used here was MinION. This is the smallest device, and number of reads that you can get is, I don't know, from 1 to 5 million reads. And for example, I will give you just kind of overview of for our samples and our experience. So we use PBMCs. They are not super activated, so you don't have many poly A tails. So, and we started from around 1 microgram of RNA. At the end, we managed to found when we do sequencing, when we perform sequencing, we end up with around 10,000 genes that were annotated, and around maybe 200 were differentially expressed. So, and the average reads was around 2.4 million reads, basically. Now, next instrument is PromethION. and this uses a different flow cell that has many more pores, and it can go from 10 to 30 million for direct RNA sequencing. So it's much more, the instrument can give you so much more, and of course you can go much deeper. And I think even here, even if, to come back to your question, even if we cannot compare depth directly between two platforms, it is evident that with greater depth you can reach again targets that are less abundant. So if you are again seeking to use direct RNA sequencing for analysis of long non-coding RNAs that are relatively low expressed, not all, for of course MALAT, NIAT, and those one are also super present in our data. You can find them very easily. They are one of the highest expressed genes, at least in PBMCs. But if you go for some others that are relatively low expressed, then MinION technology, I don't think can work for you if you don't do some enrichment first and then do the analysis.
Okay. Okay, great. Any other questions?
Maybe I can ask you one question, Milan. It's not so much about the RNA sequencing, but well, it is. But it's a problem that we often run into, is that as soon as you have your RNA-seq data and you do the analysis and you find some really interesting targets, and then you're like, okay, let's grab qPCR and see how well that matches. And then it really doesn't.
Yeah.
Do you have any thoughts on that? Do you have any, I mean, I know it's a general issue, but do you think
you are just asking me this question at the exact same moment as I'm currently designing primers for my targets that I found in my data, and I will come back to you in hopefully the end of January, beginning of February, when we validate this on the entire cohort to say this goes and this doesn't go. What can I tell you for sure? So far what I see, because I read a lot of papers lately to see what other people found. We are currently the first one who performed direct RNA sequencing on PBMCs in patients with myocardial infarction. And I look some of the genes that were dysregulated in our data and compared to some other data sets from PBMCs from other, but done with Illumina. And I can tell you that a couple of those genes are matching. Not all, but so I have around 200 differentially expressed genes. Let's say from top 10 candidates, I found 5 genes that are already published in other data sets done with Illumina. So based on that, I can conclude that at least there is some consistency in the literature. I mean, you know, from me to literature, I cannot tell you from this point how it's going to be when I do the analysis. We chose around these 10 genes to really do to try to validate and see what is going to happen. Regarding M6A, that's a completely other story because validation of M6A, currently we found only one paper how you can do combining 2 different reverse transcription enzymes. One is lazy, that basically got stuck when it sees M6A, and the other goes through M6A, and then you compare CT values just to see if one reverse transcription is more efficient to the other, and then you conclude, okay, on this spot there is an M6A, and we plan to do this method. We are currently optimizing it for its use, because in our data we can see, as I mentioned, M6A on specific positions that are certain positions that are differentially methylated. But for example, in my case, what the top genes that are differentially methylated have 30% of difference in methylation. So the question is, will we be able to validate, you know, if the method for with qPCR is sensitive enough to pick up the difference of this 30%? And again, I cannot answer this right now. We will do this in the next several months, and then I will, of course, give you the feedback on this, because I know that you are also interested. in that. But at least this is something that I don't know how to interpret, but top genes in terms of differential methylation is 30%. So nothing more dysregulated in terms of more than this 30%.
Thank you so
Okay,
much.
do we have any questions from the audience, or they are completely puzzled and even more confused? Hope not.
I think they are enlightened and evening.
Yeah, hopefully. In any case, if anybody has any questions and just they are shy to ask, please drop me an email and I will try to give you some profound answer. I'm not certainly an expert in the field, but it is quite challenging when you want to do RNA sequencing, not only from the perspective of not having enough RNA to do whatever we want, but also from the perspective of what type of approach you will use to prepare your library. Are you going to do the polyase selection, or you are going to use ribosomal depletion and all the other things that I mentioned? So these are all the things that you really need to consider prior you engaging with RNA sequencing, and that's the best advice I can give you. And just close your eyes and hope everything will be okay at the end. Yeah, so that's it from me. So I don't know if there are any other questions regarding the subject. No. Okay. If there is not anything, then we are finishing for this year. I would like to thank you, my panelist collaborators, Paolo, Dimitris, and Yael, for hanging out during these sessions. I know we are all super swamped with all the other things that we need to do, so it's not so easy to always dedicate time for this. So thank you. And also to all attendees. I think were quite successful this year. So hopefully next year we are going to start in January. Currently, I didn't came up with any plans. I will go through. We have database of people who applied for January and February, and I will try to contact them and see who is going to be available to give specific to address specific topics. So by the end of December, I think I will schedule the next webinar for end of January, probably around 20th of January, and then you will get the information. And with that, from me, I will say goodbye. I wish you all the best in 2025. Have a nice holiday season. Enjoy, relax, don't think too much, don't work too much. We need sometimes to relax and really focus on other things. I hope that everybody has nice plans during the holiday season. And I really wish you all the best. Enjoy, and see you again in January.
Thank you, Milan. See you next year.
See you. Enjoy. Bye-bye.